Good morning, good afternoon, good day. My name is Peter Dahl. I am the Deputy Director in the Vaccine Development Function at the Bill and Melinda Gates Foundation. Today I'm going to be reviewing correlates of protection for COVID-19 and talking about what we've learned so far. My other hat that I wear is I am the convener for what's called the COVE clinical SWOT. We've put on a number of workshops over the last 12 months or so with partners at CEPI, and within that clinical SWOT, we have a working group on correlates of protection, chaired by Stanley Plotkin, and a lot of the work we're talking about here today was in part aggregated with help from that working group. And as we've been moving forward on trying to evaluate and explain and understand correlates of protection for COVID-19. The importance of correlates of protection are well understood, but also important where you are and what you do on the vaccine development journey, or from a policy or discovery standpoint. This is a slide on the left that Stanley Plotkin has shown at a few. He gives great talks on correlates. You should try to find one. He's done it at WHO. He's done it at some of our workshops, but just sort of enumerates the basic points around correlates of immunity and what they help us with: basic immunology, enabling correct choice of the vaccine antigen, from a manufacturing perspective, consistency of the potency of the product, on an individual level, determining susceptibility from an individual or a population level, from a product developer perspective, moving from a first generation vaccine, say a canonical Wuhan-based vaccine, to a variant vaccine, to a second generation. This is correlates are critical for that move. And finally, something that we've been wrestling with for the last number of months with COVID is, as efficacy studies become either infeasible or unethical, immunologic data are the only way we're going to be able to license new vaccines. So on the right, you kind of see the different stakeholders. Whether you're a discovery basic scientist on the immunogen design, correlates are important. Trialists and regulators. As a regulator perspective, I mean, how do you communicate the criteria for study success? Lacking in efficacy, what should be the comparisons? What should be the endpoint? What should be the success criteria? All rely on having a good understanding of correlates. Manufacturers, lots of scale up, lots of site movements, tech transfers of products during COVID. There's always going to be differences in the product that comes from each of these steps, but do they matter to the safety or the efficacy of the product? So understanding around what a correlate is and what it would be is really critical for us all, regulators in particular, to say, hey, this product can also be used despite efficacy not demonstrated from this precise product. And then finally, policymakers, whether you're at the JCVI in the UK, the Antibody in India, or the WHO SAGE, you are going to be confronted with questions around declining titers or variance or immunologic readouts that you need to say, does this matter? Do we need to institute additional dose? Is there going to be an efficacy concern here? Knowing what these correlates mean is critical from a policy perspective, and that's a hard one, but we gotta get smarter. So what's in our toolkit for making building an understanding around correlates? And this slide could be representative of any pathogen: a meninge correlate, a norovirus correlate, or a COVID-19 correlate. We kind of draw from different elements in the tapestry of data to inform whether a correlate is present and what is the biomarker that mediates it. On the left, you see, vaccine-induced immunity. That's the one we'll talk most about today. And using phase 3 efficacy studies, either on population-based immunogenicity or individual breakthrough disease analyses are two tools that we use to give us a steer on correlates. Natural history of disease, we saw this early in the outbreak, longitudinal reinfection studies, healthcare worker studies from the UK and other places looking for titers prior to a new onset infection and trying to characterize those titers and that immune profile. And then passive immunization. Often these are done with either monoclonals or convalescent sera from surviving either animals or humans, and seeing what protects either in an animal challenge model. Imperfect, but it's a steer, and we saw data I'm not going to talk about from Dan Baruch and others early on that gave us a good idea on this. And then there are other sources. Some are more relevant than others. Challenge CHIMs have not borne fruit yet, but in other products, typhoids or Shigella, those may be more relevant. PrEP studies of post-exposure or pre-exposure monoclonals or convalescent sera. And then importantly also disease among immunocompromised. So if you don't have B cells and you get bad disease, maybe that tells you something. If you've got HIV and you've got no CD4s, you get bad disease, that's another indicator of what is the mediator of protection. So all of this comes together as a tapestry of data to guide where we're at on correlates. So let's go back in time a little bit. So back in the first glimpse around correlates that we started to get, this was kind of a fun one a little bit, but it was a fishing vessel in the Pacific Northwest of the US in the Puget Sound area. They swabbed and got blood draws from all 122 people on the ship. Interestingly, just 3 of them did have neutralizing antibody before getting on the boat. Somehow one snuck through with COVID, and there was a large outbreak, 82% attack rate on that ship. And interestingly, all 3 of those individuals with neutralizing antibody were among the 18% that did not get disease. So it's a whiff of interesting data that was suggestive, but certainly just anecdote. We started to get better data soon thereafter. Here's the study from the UK, from the ChAdOx vaccine, phase 3 study. As you may recall, early days, it was suggested that there may be a 1-dose vaccine. Eventually, it became a 2-dose vaccine in the phase 3 efficacy study. As a consequence of that shift, we had an interesting situation where those who got 1 dose and then later they decided it was a 2-dose had a long interval between doses. So 12-week interval for some, 6-week interval for others. And they had enough numbers that they were able to do interval specific efficacy. So here's the narrow interval, lower neuts, lower efficacy. The 8-week interval, lower neuts, lower efficacy. 10-week interval, higher neuts, higher efficacy, widest interval, 12 weeks, highest neuts, highest efficacy. So as the interval between doses increases, neutralizing titers increase, efficacy point estimates increase. That's a good story. So another glimpse around the value of neutralizing antibodies during protection. Then kind of a gold rush of efficacy studies came out toward the end of December and into January and February of 2021. And so we had efficacy from Novavax, from Pfizer BioNTech, from Moderna. We call these sort of the high efficacy products, between 94 and 95%. The Oxford one we just spoke about, but Sinopharm read out from the UAE and around 80% efficacy, and between 60, 70, 80% efficacy from the AZ Oxford study. And then we had Sinovac, which ran the study in Brazil, had modest efficacy, acceptable efficacy, 50%. And what was pretty clear, although the titers were all over the map when you controlled for the titers using a human convalescent sera panel, so a ratio of the neuts to a human convalescent sera panel, and these are imperfect, but everyone did have a mix of moderate, severe, and mild disease. You saw the ones that were higher, neuts versus HCS, gave the higher efficacy, 3- and 4-fold higher. The ones who had comparable neuts versus convalescent sera, comparable, modest efficacy, and Sinovac was the outlier with actually much lower neuts than their convalescent sera that was reported lower efficacy. So that was, again, very interesting and instructive in early Q1 of 2021 that neuts and protection were related. So, I mentioned the human convalescent sera. This is just a graph from the Earl et al. Paper to if you didn't normalize with human convalescent sera and just looked at the neut titers and tried to run this same graph of efficacy on the y-axis, neuts on the x-axis, it's a scatter plot. Same thing with binding, efficacy on the y-axis, neuts on binding antibodies on the x-axis, and it's a scatter plot. So that's just indicative of everyone's got their own assays. They use different reagents, different standards. It's hard to compare. So when you did compare them then using a standardization, a poor man standardization with human convalescent sera, here is the data with the binding, with a rank correlation of 93%. Nice connection to the line, and here are the neuts with a similar high rank correlation of 0.79. So a strong relationship between either neuts or binding versus efficacy. And the colorful boxes here are just meant to indicate that this holds firm across 4 different vaccine platforms: inactivated vaccines, ad-vectored vaccines, RNA vaccines, subunit vaccines. So that's a story that I think is helpful because it doesn't matter how you get those neuts or binding. It doesn't matter what the road you take to get them; they all indicate and are related to efficacy. So this was, again, a strong indication that these are important. Some additional analyses done in the Earl paper, again, is just to look at if we peel out from the J&J and just look at the Wuhan strain, D614G, that 66% efficacy. If you just look at the D614G, that becomes a 72%. So it moves closer to the line. Novavax, that 89%, was in a mix of alpha and D614G. If you just look at the D614G or Wuhan, it goes up to 96%. So that little dot goes right on the line. That little dot goes right on the line. So if you peel out and just look at Wuhan, it's even the relationship is even stronger. So just again, reiterating, I think that there is a strong relationship with symptomatic disease within 2 to 4 months after vaccination and neutralizing antibodies against the parental canonical. All those caveats are important because that's the window in which we're talking, not going beyond that for now, but that's important. So that is the paper I just talked about. It was the Earl paper. So it had both neutralizing as well as binding and that relationship, and excellent analysis was done as well by Corey et al. In Australia. Got data from Covaxin, additionally also convalescent sera and some other predictive models, which was really a well done paper to use to, again, emphasize the relationship between in this case, neuts and protection. So where does that get us? The first problem, back to this journey and what you're trying to use correlates for. I think these set of studies have been important to help regulators and trialists build the evidence package for new vaccine registration pathways based on neutralizing antibodies in the light of the challenges with the feasibility of new efficacy studies. It also reinforced, I think, the guidance that we had very early from regulators around moving from the parental strain vaccines to variant vaccines, let's call them first generation to second generation, based on comparable new titers. And that's FDA, EMA, various regulators signed on to that one very early. But the cross-platform analysis is something that we're still on the way to. All right, moving on. So the next problem, I mentioned that there's other stakeholders for correlates, and the other one is the policymakers. So how do you translate immune response data to recommendations on boosters, on immunocompromised? So this, we're often talking about how do they inform individual level protection. So if grandma's got a titer that's down in the 30s, does grandma need another dose or not? That's an individual conversation about an individual subject, and how do you make policy decisions? That's not licensure. That's not how do you get a new vaccine that you have confidence will work through. This is around making determinations of susceptibility on individual populations. So some new data kind of to inform that comes from the breakthrough analyses that Peter Gilbert presented at the WHO R&D blueprint consultation. So Peter Gilbert's at the Fred Hutch, is one of the statisticians, is part of the team that has been analyzing all of these Operation Warp Speed studies from the US government. This was the Moderna study. With breakthrough analyses there, we should expect J&J, we should expect AZ, we should expect Novavax, each of those funded by the US government. I don't know if Pfizer ever will, but Pfizer certainly could do this as well. But those 4 studies will be coming forth. 30,000 subjects randomized 1-to-1, 100 centers. Importantly, sera obtained from all subjects at each time point. So day 1, 29, 57. Sera only, no PBMCs. So CMI is, we can't study CMI from breakthrough analysis. Hard to process that many B cells in 30,000 subjects, but that's what we're looking at. So Peter Gilbert was able to present data on this breakthrough analysis comparing individuals who went on to get breakthrough disease despite vaccination against a large group who did not. Here are the spike and the pseudoneut results. Let's just focus for now on the day 57, so 4 weeks post dose 2. These are the 36 subjects who got 2 doses of Moderna vaccine and then had symptomatic COVID disease at least 28 days after the second dose. Maybe it was 14 days, 7. I can't remember, but at any rate, you can see here that the titers were modestly lower if you had breakthrough disease versus if you did not have breakthrough disease. So those are statistically different and sufficient to do an analysis. But importantly, also, you have a number of subjects that had high titers, reasonably high titers, up to 1,000, and still had breakthrough disease, something that is not completely surprising, but is something that to bear in mind as we're talking about an individual level protection. So Peter and company went on to analyze this data to generate a curve which shows the predictive efficacy based on a given pseudoneut titer here on the x-axis. So just take an example plot, let's say 80%. If you want 80% efficacy, what level in the post-Moderna vaccine would you need? Something around a pseudoneut ID50 of around 20 or so. Same thing was done with the IgG curve as well. Less robust data, you can see the distribution of titers. They're much tighter distribution of titers, whereas with pseudoneuts you have a broad shoulder of titers, which gives you a better and more confident curve around the pseudoneut assay. So that was great. Further confirmation that there is a relationship. Additional breakthrough analyses were shown by Maren Voisey at the University of Oxford University, who analyzed the AZ ChAd breakthrough analyses, and that's here on the right side. Here again is the distribution of neut titers after 2 doses of ChAdOx, and she was able to generate this distribution curve. Again, let's just take 80%. You run it across, look at this line, you drop it down, you got a pseudoneut titer of around 40 units in international units. And what Maren was able to do was then to take and overlay the ChAd data, because it's thankfully using the international units, with Peter Gilbert's Moderna breakthrough analyses. And here, this little green curve is the ChAd data, and right above it in the dotted line is the Moderna data. So you can see, and she was able to, plot them against each other. ChAd, 70% efficacy, titer of 8, 90% efficacy, titer of 140. And that's remarkably close to what Peter Gilbert was able to generate with the Moderna. So importantly, I think this is across 2 platforms. This is an ad-vectored vaccine. This is an RNA vaccine, and both giving with a independent breakthrough analysis from large efficacy studies, comparable, in this case, pseudoneut titers that predict a comparable level of efficacy. So we like it when these things happen, and 2 different independent analyses give us roughly the same outcome. So lots more work to come. Lots more analyses, as I alluded to, other breakthrough studies that are coming. But where do we stand now? I think there is a general consensus that neutralizing antibodies, IgG to spike, appear to predict protection against symptomatic COVID disease. So that's short term. That's Wuhan. And it doesn't matter how the antibodies are generated, whether they come through an inactivated vaccine. You saw the COVE vaccine from India is on the line. The ad-vectored vaccines are on the line. The RNA are on the line. So even convalescent sera, those antibodies are predicting similar levels of efficacy. Didn't talk about this much, but the pattern of protection appears to extend to variants. We do see reduced neuts and IgG in the presence of lower efficacy against symptomatic. So the way I would characterize it is the level may be different, but the mechanism is likely to be the same. We do need those analyses to be done, and there are some work in progress around at least binding antibodies against the variants and showing a relationship with efficacy. More to come on that, I would say. I think this is an objective truth. Several national regulators, not all, have indicated support for new licensure of new vaccines through immunobridging. So MHRA from the UK, we mentioned, but also others in Korea, India have indicated support for immunobridging to support new products for efficacy. Importantly is really around severe disease. Is there a level that is protective, or is there a different mechanism that is protective for severe disease? Is this where CMI and CD8s are more important? Durability, those data will come, but I think, as I said earlier, really those neuts are short-term neuts. And this is where I think we have to continue to lean on effectiveness studies to guide booster decisions and not just look at declining titers. And I think that's the scientific consensus. CMI, other antibody functions, FC effector functions, not quantified. We think we may be able to look at future meta-analyses from smaller studies where we've got well-characterized CMI in the same lab using the same process across multiple vaccines. This is where I think we're hoping toward the end of the year we'll be smarter about the relative contribution of these other parts of the important parts of the immune system. And then finally, assays. We need qualified, validated assays are critical. Please, everyone, use the international standard. That's a global good so that we can talk across these different studies. So with that, thank you for your time, and that concludes my talk. Bye-bye.